the that TVK Singh joined from Hyderabad. Welcome, sir. Right.
Dr. Singh, good morning. He's muted.
Yeah, please unmute, sir. That TVK Singh, sir. Yeah, you go to sharing directly. First sharing, na? Be here. Share. Okay, no? Okay. Yeah, this recording. This one recording.
Start ಮಾಡಿದ್ವಿ.
There you started.
Start ಮಾಡಿದ್ವಿ.
Okay. A very warm welcome and good morning to one and all who joined. Dr. Varghese, Dr. TVK Singh, today's speaker, Dr. Joseph Rajkumar, and all others. Before the talk, I have few things about the milestone what we have reached in the endeavor of sharing this knowledge in plant protection in horticulture, with particular reference to horticulture and agriculture in general. We have reached 125th month. This is the 125th month after we started this in August 2013. And today we'll be having 160th talk. Before that, we made an attempt to combine all the talks, what happened in the first 10 years, a decade, that is 155 talks. Here, what we are seeing in the slide, I hope it is visible. Sir, is it visible?
Yeah, it's visible.
Okay. This is a Aapme Plant Protection Talk Series. This today's specialty is, today is going to be the 125th month's talk. That credit goes to Dr. Joseph Rajkumar, and we will be giving a talk on exotic whiteflies and with particular reference to their management using biological control agents. Coming to the today, we have come out with an abstract book which contains details of 155 talks till last July, up to July, where we have completed 10 years, that is 120 months. The speakers in the last 10 years, they include many professors, ICAR system, faculty as well as students, and scientists from USDA, ECP, Kenya, World Vegetable Center. One talk, 100th talk was through scientists from Mexico. That topic was on SIT, and several speakers from SAUs, one Indian Institute of Science, then one Indian Administrative Service, one IAS officer was called for celebrating plant protection in horticulture crops. Then some professors from engineering colleges, some speakers are from plant protection related companies, and students, faculty from both ICAR institutes and SAs. The whole lot of speakers are there. Out of these 155 talks, there were more than 110 speakers who gave and shared their knowledge on plant protection. And today we'll be sharing with you the abstract book, which is open for download, and we'll be putting this in Zoom only, and it will be circulated to in all Aapme members and others also, whoever is joining. It will be sent to Aapme group mail also. Anyone of you who are interested to give a talk and be a part of this endeavor, please mail your interest to the aapmhe@gmail.com. We are open in a month, 1 to 3 also. We can, even if it is a small topic of 15-20 minutes. You are welcome. If it is a new one and you are ready to share that knowledge, you are welcome. And you can be a part of this endeavor. Coming to this today's what we have compiled, AAPMHE, a decade of monthly talks on plant protection. This is a small abstract book, 120 months and 155 talks. We made this small compilation, which will be shared with all of you. This is coming to the, I will be giving a link through this Zoom in due course. Now we will go to this today's presentation. This unsharing. Just unshare this. Not unshare. Yeah. Coming to today's topic, which is going to be the 160th in the series, and this is going to be the 125th month. The topic is on exotic whiteflies on palms and biological control. Today's speaker is Dr. A. Joseph Rajkumar, who is coming to the brief introduction about today's speaker. Dr. Joseph Rajkumar is currently working as principal scientist in entomology division at ICAR Central Plantation Crops Research Institute regional station located in Kayamkulam. He has completed his UG and PG from TNAU Coimbatore and a doctorate from Indian Agricultural Research Institute, Pusan, New Delhi. And he has undergone a postdoc under DBT, a fellowship at SPIC Science Foundation, Gundi, Chennai. He had worked as assistant professor at Cardamom Research Station, Kerala Agricultural University initially during 1999 to 2007 before joining ICR. In KAU, he refined IPM technologies for cardamom and black pepper and was instrumental in the release of 3 cardamom varieties, namely PV2, PV3, and PV5. Subsequently, he joined ICR Central Plantation Crops Research Institute, Regional Station, Kayamkulam, Kerala, during 2007 and developed novel botanical formulations as pest management solution in coconut and validated coconut-based ecological engineering for pest regression through stimulo-deterrents. He has discovered a sooty mold scavenger beetle, Leocrinus nilgrianus, involved in bioscavenging of palm leaflets infested by rugospiraling whitefly, which is an example of recent exotic, and he has established conservation biological control of exotic whitefly. He has identified and characterized 2 invasive nesting whiteflies infesting coconut and reported their occurrence in India for the first time. He has published more than 70 research papers in peer-reviewed journals and presented several papers in national and international symposia and authored a book also. Coming to his peer recognitions, he has received several national awards, including ICR Jawaharlal Nehru Award for Doctoral Thesis, ICR Lal Bahadur Shastri Young Scientist Award, Professor T.N. Anantakrishnan Award for Senior Scientist for the year 2018 and 19, and Azra Young Scientist Award. I'm also happy to share that he is a life member and fellow of our association, who is organizing this series of seminars, that is APME, Association for Advancement of Pest Management in Horticultural Ecosystems. And he is also a fellow of ICR Society for Plant Protection Sciences, New Delhi, as well as ICR Entomology Society of India, located in Entomology Division, New Delhi. Yeah, with this brief introduction, now I request that Joseph to go ahead with his presentation on whiteflies and biocontrol. Over to Dr. Joseph Rajkumar.
Yeah, thank you, Dr. Sridhar. Can I share my slides now?
Yeah, please go ahead. Now, complete sharing is in your hand.
Okay, thank you.
Okay.
So my slide is full now?
Yeah, it's visible. First, clear.
First slide is fine. Okay. Fine. respected Dr. Sridhar, the president of the APME Association, Dr. Varghese sir, Dr. Singh sir, and my dear friends. I have Dr. Ansar Ali also on the screen. And it's very nice that I have been selected for this very unique talk, which has been regularly happening in this societal website or in the societal position. And I am always very keen to track it up, because I should give big accolades to Dr. Sridhar, because once he has taken over, this has become so vibrant. And the resultant is what he has just projected, a kind of an abstract book as well. So I'm very happy. I'm very thankful to the team, especially Dr. Sridhar, for having given me this wonderful opportunity to share some of my experiences on these exotic whiteflies occurring on coconut in particular, and the exotic whiteflies in general, and share my experiences maybe for the past 5 to 6 years in this line of research. So coming to the topic per se, exotic whiteflies in biological control, as introduced, I work under the ICAR CPCR regional station at Kayangulam. So Kayangulam is just in between Trivandrum and Cochin, at least for those who are not aware of where we are. And we are working on coconut diseases and pests. Our main concern is on the root wilt disease. And once the palms are a bit affected by the disease, what happens is it becomes more susceptible to the insect pests and other nutritional disorders as well. And to be very frank, our institute has a very past legacy to highlight with. I will share some of my experiences about your institute as well, because my talk will be around some brief introduction, something on the whitefly diagnosis and diversity, which is very important, because unless you identify that, it becomes very difficult to correctly manage that as well. And we have excellent biosuppression strategies, as well as we have one beetle which is responsible for bio-cleansing, a kind of cleaning the palms infested by this whitefly, especially on the sooty mold. And we have a kind of a crop habitat diversification system, a kind of a heterogeneous landscape, which is very important, because once you have the diversity, in what way we can reduce the pest incidence in toto. So that will be one of the areas where we link that with a kind of a One Health approach as well. And finally, on a conclusion, the way forward. So coming to my institute, because I am really very fortunate to be a part of this very great institute, because it had a humble origin way back in 1947, which means last year we had our Azadi Ki Amrut Mahotsav as well for our institute, crossing 75 years. And it was the foundation stone was laid none other than His Highness Marthandavarma Raja on 24/4/1947. So that was the nature at the time, and we were part of the biological control series as well, maybe in 1970s. So that goes to say how old our institute is. And last year itself, we conducted a series of program, because our emphasis is mostly on the biological control. We have a legendary past. We have Dr. K.K. Nurla, Dr. Chandi Kurian, Dr. G.P. Pillai, Dr. Sadyama, Dr. C.P.R. Nair, Dr. Chandriman. All my predecessors are great legends in their field of work. And we had a kind of a block last year. We have put in some 75 coconut planted together to commemorate the 75 years of our institute. And we also had one India Post cover highlighting the superiority of our institute. So this is just to say the origin of our institute way back in 1947. But before which, I would like to say this institute institute took shape from a kind of a project funded by Imperial Council of Agriculture Research way back in 1937. So that's again to highlight, because at that time, the fund was for 25,500 rupees. So that shows how even the ICAR, the Imperial Council of Agriculture Research, was interested in our institute way back in 1937. And there was one Dr. M.K. Varghese who championed this particular program way back in 1937. And at the time of getting a grant of 25,500, one sovereign of gold was just 30 rupees. So that shows how much of importance our institute had at the time. because coconut was doing very well, and the coconut is supposed to have an international trade to a greater extent at that time. So we had an international central coconut committee. As you all know that coconut as such is a wonderful ecosystem service provider. It is only because of coconut we have the islands intact. So that is why we are very proud to say about even a particular state called as Kerala is named after the coconut as well. So the culture, the cuisine, and the custom associated with coconut is so vast. So even if you see the number of the birds, even the insects, it's a kind of a biodiversity hub. So that is one of the reasons why even this is offered to Almighty these days is from the tradition. So that shows the role of this coconut as a crop in terms of the ecosystem services, as well as in terms of the environmental well-being. So working in this crop and working on some of these pests and characterizing this makes us very, very happy and proud of it as well. So I will just highlight, because once I say about now these days it is ecosystem service provision, and the major threat is being faced with the climate change, and the climate is not now changing, it has become a climate extreme. So the major outcome of that is nothing but a kind of a biodiversity decline. So how to recoup that? That is exactly what we have to think about. So if you just see, around 75% of the genetic diversity is being lost in the last century itself. So this indicates we are focusing only on very few crops. Maybe the agriculture is presently dominated only by around a dozen crops. What happened to the others, we do not know. And you can just see the diversity of the maize itself. See the color, see the shades of that. And again, the dominance in the group. We have only around 4 crops yielding more than 50% of the total production. So is that the right way of agriculture? So that is exactly where we have to think about. And once you lose the biodiversity, the resultant will be a tremendous impact on the entire system. And that is one of the reasons why you find this invasives taking a toll in these areas. So I would just say that we are knowing about very few. We know only about 1.7 million species of insects or plants or animals out of the 100 million, which is unknown. So we have tremendous scope to go into that. So once you understand all these things very critically, we will be better placed in terms of the services which the insects can do and other things. See the diversity in coconut alone. We have a very small coconut to the largest coconut. See the shades of the coconut. We have the different colored. It has a brown colored, yellow colored, brown. All these diversity encompasses everything, and that is where we have to give focus these days, rather than going a kind of a system where we focus only on a few varieties as such. Now, invasive pests has become, even during the initial talk, Dr. Varghese sir was highlighting what exactly is the reason why these invasives are entering the country. It is mainly because of the increased trade, tourism, and a kind of a travel which these days are happening, no doubt about that. And also we bring in lot of these materials unknowingly as well, because most of these ornamental plants or ornamental palms are, but for that matter, anything which is interesting abroad, when a person visits, he tries to bring it into our country, and our quarantine are not that stringent to see that it is not being introduced very successfully. And the simple reason here is once it crosses the native biodiversity location, it doesn't have that kind of the natural enemy complex to suppress it. So that is one of the reasons why it outsmarts in the breeding processes. It threatens the biodiversity and the very human livelihood as well. So the simple reason being it breeds profusely in the absence of these natural enemies. So this is a very important threat of late we are being faced, next only to the climate change, because the invasives are creating a lot. So in palms, which is very perennial, which is quite robust, it can withstand the pressure of invasives to some extent, no doubt about that. We had the introduction of Batrachitra arenosella way back in the 80s. It never caused any greater problem. It was there, and we had excellent natural enemies in the system, because as such, coconut is not being suppressed with a lot of application of insecticides. And we had even Meloceros undulatus, which was reported in 2010. And that again never exceeded beyond certain limit. It was restricted mainly. You can see the damage by this Meloceros, just like a kind of a notching kind of symptom. And here in kind of Batrachitra arenosella, we'll find the flowers webbing to especially the main flowers. And of late, in 1998, I should say, it was the coconut idiophyte mite introduced in the country, and it is from our own laboratory. Dr. Satyaman team in 1998 could discover it for the first time, and they have reported it. And you know the outcome of that. Between 1999 to 2005, lots of international projects have happened, and the entire outcome of this is just the advice to go for a kind of pesticide holiday, and they promoted mostly the bioagents, especially the neem-based products as well as the Hirsutella thompsoni, which was taking. And now even in certain time when there is a shift in the monsoon showers or there is a shift in the weather parameters, we find the mite is shooting up, and 2023 is an example for that. So what happens initially when a invasive enter into the country? It can be through the transport mechanism, or it can be a deliberate attempt of introduction as well. And once it introduced, it is having a kind of a check by the natural means which are present in our system. And some or many may not work as well. But one classical example which took us in surprise is the Rugi operculatus itself, Alerodiscus rugi operculatus, the rugospirine whitefly. We had a previous introduction of Alerodiscus dispersus in 1993, and the final stages of that, especially in 200, we had the introduction of the Encarsia. Encarsia was introduced from our Lakshadweep Island, so that was prevalent in our system. So thanks to that introduction part, I think Dr. Remani, Dr. Purni, were all involved in this process of introduction from this Lakshadweep Island, and the resultant is it could bring down the population of Alerodiscus dispersus, and we also had the Rugi operculatus population also tremendously. Otherwise, this would have become a real threat to that. So the introduction part becomes the first phase of that, and then the establishment happens. Establishment will be more successful if the natural means are absolutely lacking. So that is one of the phase where we can have a slight check. And the third one is the abundance of the shooting up of the population. So that is a very important area when the natural means are not lacking. That is the phase where we can very clearly introduce have a kind of the classical biological control, taking care of the natural means from the other areas where it was introduced. That is why whenever an invasive pest comes into a country, they will all look for from where it has come from. So once you know the center of its origin, naturally they will have the natural means in that particular location. So we'll be in a position to bring it out. And that is why the biodiversity and the biocontrol makes a very important thing. So before it has a kind of a disposal, now I think in most of the things, it is kind of a flattening thing. So we have to intervene before that phase so that it is not creating a kind of an incursion level, or the invasive potential is not increased to a great level, for which timely intervention, suppression, either through the nature-based solutions are very, very critical, and for which coconut is one of the classical example which could withstand all this barrier. Coming to a brief history about this whitefly research in our country, I think it was Linnaeus who has misnamed whitefly as a kind of moth. He has named it as Tinea proletella way back in 1758. That is how it has started. And it was immediately Lapel who identified that it is not a moth-like organism. It is definitely a hemipterous insect. So he brought it under Alerodus. So Alerodus, as you know, aileron comes from the bird floor. Floor is just the whitish powder with the wings produced. And it was Westwood in the year 1840 who could make Alerodidae as a family per se. So that is how the whiteflies started to be named after. And in 1895, it was Maskell who has reported some Indian Alerodids as well. So he's the first person to do something on the Indian Alerodids. In 1903, we had PL published a paper on these Oriental whiteflies. So these are all very basic information about the Indian whiteflies exclusively done by Western persons. And the classical persons, I think a paper which everybody should go through is the Quantons and Baker way back in 1913-14. It describes about all these whiteflies in a very systematic manner. Some of these line diagrams are simply classic, so they have to be well taken. Those who are interested in this whitefly taxonomy or whitefly research for that matter. And from India, it was Mishra in the year 1923. He has reported Trialerodus ricini from the castor. It was named after his kind of contribution. So that was the first beginning, I can say. But thanks to the efforts made by Professor Karam Singh, I think he worked with the Pusa Institute and he has done a tremendous work. I think even the present day generation could not follow what he has done at the time in 1935. He has published a kind of a memoir. I think all taxonomy should go over this particular memoir, which highlights almost 44 species beautifully pictured by line diagram. So there will be no confusion at all. So any whitefly worker should go through this particular work by Professor Karam Singh way back in 1931. At the time, the print, everything, you know, what kind of difficulty they but beautifully he has made it, in which he has described some 25 new species as well. Then it was in 1945 by Hussain and group, they worked on this cotton whitefly, Bemisia, and Usman and Petraria, they also done some works on the Karnataka whiteflies, and Dr. Rao, I think in 1958. So these are all some works afterwards, which was not well known to the people or well known to the scientists, but it was only Professor David, B.V. David, was started very systematic work. I think I give a big salute as well, because it was his effort which has made the whitefly taxonomy more vibrant in the ester years. I think he has very good students, which includes Dr. Sundaraj himself is one of his students. We have a team, Dr. RSW, Jesudas, and all these people, including Dr. Subramanian, all they have done tremendous work. And finally, we have a very good team, very classical experts available in the country. Even B.V. David is still there, and two of his followers, I think I should give great credits to Dr. Sundaraj, who has retired just now from Indian Institute of Wood Science and Technology, and Professor A.K. Dubey. And they are all, Professor Dubey is in Zoological Survey of India. I think because I have not mentioned any of these papers of Sundaraj sir and Dr. Anil Dubey, because everything is now available in the Google Scholar if you just go through. And they have made whitefly a taxonomy very proud even at the international level. And we can be really very proud of this contribution by all our Indian experts. And whenever even the foreign experts now have any kind of clarification, they'll immediately have Dr. Sundaraj or Dr. Anil K. Dubey to have their own clarity, because it's something very great which you have to do. So something about taxonomy, I think it is a well-known. Just put it in because it is the puparium which is used for identification. That's very important. And also now adults are involved in the identification process. And finally, you will have even the molecular taxonomy, which is very, very important. I think I'll share some of my thoughts on that as well. And what we do initially is whenever you go collect the specimens, you put it in the KOH kind for an overnight room temperature, what happens, just to take away the internal contents. And once the internal contents are taken away, put it in the alcohol for just 10 minutes or so. And the right amount of acid fraction makes a lot of difference. So that add it. And once you add the glacial acetic acid, maybe a few drops of it, it will see that the fixation is perfect. So once the glacial acetic acid is a very good fixative, the color will be fixed onto the puparium. Then you can view the things very nicely and rinse it in alcohol, transfer it, put it in the clovides and view under the microscope. So if you don't view under the microscope the puparium, we become a big zero of that. So unless you see the puparium, identify the features perfectly, and then classify ourselves, we make the things very great. So this is something about the major. I think these are all available in the literature. We need not do. So the main point which I wanted to highlight here is about the vasiform orifice. So this is the area I think the whitefly identification makes a lot of extent. And we have the submarginal fold here, because this is a submargin area where some of these CDAs are present, which are very important. And to be very frank, this vasiform orifice has a flap-like structure at the top. So this flap-like structure is called as operculum. So vasiform orifice contains two parts. One is operculum, another is a kind of a knob-like structure, which is called as a lingula. The shape of the lingula, the shape of the operculum, the features in the operculum, and some of these compound pores, which are normally present in Aleurodicinae, are all very important in the exact identification of the species. Okay, so the species identification is very important, because once you identify the correct species, then comes the exact management strategy. Because as a pest managers, we'll be very happy to manage a pest, but to know the pest is equally very important, for which a bit of taxonomy has to be put into the mind of all pest managers as well. So that's very important. So we also do the molecular characterization part. In addition, we also do the adult taxonomy, especially we study the genitalia. Because normally in whitefly, genitalia is not taken into a main part, because mostly the genitalia work has been restricted only with the Aleurodicinae group, especially the para-Aleurodous material only, they go for the study on the adults, especially on the genitalia. But I feel that because I have handled a few of these whitefly species, what I feel is even genitalia can make a lot of insight into exact identification, at least to some extent, where there are some kind of controversy erupting. So that I will be sharing at the end. And we find a kind of the molecular characterization very simple. You get the total DNA extracted by simple procedure of a CTAP procedure. It's nothing very critical. Go for the COI gene for the PCR amplification. Once you get the PCR product, get it sequenced. Once the sequences are available, you go for the blast search, align the sequences, and you'll get a wonderful tree or the species convergence, which makes very interesting. So what happens in palm system? So in palm system, as I know, the invasives are quite common here. It's not nothing new to us. We were expecting something like brontospur to arrive into the country, but thanks, we have never met the material so far. But invariably or unknowingly, this whitefly came way back in 1996. This was when the Alerodiscus dispersers were peaking around. So Alerodiscus dispersers came into the country way back in 1993. It was first reported by Dr. Palanisamy and group on cassava. So that was how the dispersers started. Afterwards, I think even Dr. Mani, Dr. Krishnamurthy and team done, including Varghese, they've done tremendous work. I think even they evolved management through insecticide, but only after the arrival of ICAR-IIHR, or now we call it ICAR-Guadeloupe, made the difference in 2000. So in 2000s, dispersers was never reported as a major pest, thanks to the arrival of ICAR-Guadeloupe from our own Minicoy region. So this is how it is. So here what happened is Dr. Prathapan reported this as a minor pest in 1996, which means in 96 onwards, Alerodiscus dispersers is an invasive whitefly, was reported as a pest on coconut in 1996. It never took an upper hand. So in 2015 and 2016 came Alerodiscus dispersers. Rugi operculators. So when Rugi operculators was just taking an upper hand in Pollachi, in Kerala, we were speculative of thinking that it should be a disperser. So that confusion, that is why I always wanted to highlight to my own friends or colleagues, or even to my junior friends, to see that never be taken by that. So unless and otherwise, you know it to identify. So this is one of the, I'm absolutely not a taxonomist, but I have generated tremendous interest to know about these whiteflies these days, to exactly know, because it is so many numbers. I'll come to that number as well. So this took some time, and then we got it perfectly identified by our experts, and it was Dr. Shanas, Dr. Sundaraj, Dr. Selvaraj team has published this Rugi operculators way back in 2016, 2017. Then in that process came the coexistence of other whiteflies, especially the nesting whiteflies. Nesting whiteflies, normally they can be present in isolation separately as well, but many times they coexist. So that is one of the reasons why the diagnosis is very, very important, because many whiteflies sit together. We do not know what is the exact whitefly which causes the issue. And that is what Dr. Sridhar was also highlighting, to say how to make it correctly identified. That makes a difference. So we had in Bondari and Paralerodus miniae reported from Kayangulam only in 2019. And you just see here, Rugi operculators is around 2.2 mm in size. This is only around 1 mm in size. So that's the difference between these two. And you can see Bondari has a typical X-shaped marking on the adult, but adult is not used for identification, to be very frank. Here it is. So this is one of the very layman level identification for the plant. And finally, in 2019, it was Dr. Selvaraj and team, Sundaraj sir and team, they have come out with Alerotraculus attratus. And this we have reported even from Kerala of it. So worldwide, it is around 1,707 whitefly species known. So just see the numbers. And in India alone, we have around 476. And that was published in a recent book by Professor David. And of which there are 73 genera known to the country, in our country. So because, India is the hub of biodiversity, no doubt about that. So that is one of the reasons why we have a lot of these diverse species of whiteflies available in the country. And in 2003, in 2003, we reported, I think Dr. David and Manchunada reported Alero Canthus arike on arike. And it was also reported later. So this is an indigenous one. So once these invasive whiteflies have been reported in the country, this indigenous disappeared. So this is one of the reasons why we have to be very, very careful in terms of the whitefly. So you just see in our country how it has happened. So in Cochrane, we have around 5 invasive whiteflies and we have 1 indigenous whitefly. And our country, all of a sudden, you just see this is Alirhodiscus rugi operculatus. It was reported somewhere in Coimbatore, Kerala belt. In no time, it reached all part of the country. It has reached even Nagaland of late, all the island system. So we suspect it should have come from Belize, or it should have even come from Indonesia area. Indonesia and tracks were also having these whiteflies. Then we had the next one is Para-Alirhodus miniae. It was reported from Tamil Nadu, Kerala, Karnataka very severely. And now the Bontari is also reported. And especially you find this Alirhotraculus atratus now restricted. It was first reported from Karnataka. Now it has expanded tentacles even Tamil Nadu as well as in Kerala. But it is not going on the higher sides thanks to some of the excellent biocontrol agents available in the system. So though we have so many, I think I have told around 5 species of whiteflies available. The one which is still dominating in the system is Alirhodiscus rugi operculatus. Just see this, it's a bigger one. You'll find some white mottling, some brown mottlings on the wing. And it causes damage by feeding from the undersurface of the leaf, desaps the material, takes about all the content. But the resultant is it excretes a lot of honeydew. So that's a major issue. So you can see the honeydews deposited in the lower leaf of palms, and also some of the understory crops. Some of these house owners are very much worried because their cars are getting even painted by the sooty mold. So these are some of the problems which people encounter. And we have identified this particular sooty mold as Leptocyphium species. It is no more Capnodium. So Leptocyphium, the species is not identified. So Leptocyphium species is very unique to this kind of rugospilling or this Alirhodis group. So that's very important. So this is exactly why this diagnosis is very important. You just see this particular group, you'll find this is the black puparium is Alirhotraculus atratus, whereas the insect which sits here is Para-Alirhodus miniae. So when I collected samples from Mandya, I felt that I collected a lot of these adults of Alirhotraculus atratus, suspecting that this is the puparium and this should be the adult. Once I reached the Kayangulam to study the puparial features, then I was rather perplexed or confused because everything looks different now. The adult characters are totally different. So we have, unless we know perfectly what exactly the puparial features are or the adult features are, we will be rather confused because most of these whiteflies are now coexisting, especially the parallel nesting whitefly species. So this is Para-Alirhodus miniae, which is a nesting whitefly. Now it is coexisting with Alirhotraculus atratus, which is a palm whitefly, which has got a black puparium. And this has got only a light puparium, which has no convex structure at all. This will come. And this is your citrus whitefly. So this is your Alirhocanthus uglumi. And you can find this is a male and this is a female. And this is your Para-Alirhodus bontari, which is coexisting here. So don't get confused once you have this. This is nothing to do with this colony. This colony belongs to Alirhocanthus uglumi, but you will find bontari very closely associated. And this is Para-Alirhodus bontari. This, just see the size. This is around 2 mm. This is around 1 mm. You can just see how they are coexisting. So they get some of these signals from both of them. They coexist because some of these bacteria are a bit associated with what we understand. We are just started working on that particular point as well. And this is Alirhocanthus aricae. This is a native whitefly of coconut. And you'll find the other one which is very closely linked now. So this is Alirhotraculus structura, which is called a solanum fly. And in all solanaceous material, you have chilies in solanum, everything. It was very seriously attacked in 2017 till 2022. Last year, it has come down. So this is one of the indications why some species are getting displaced. So this is again Para-Alirhodus bontari. And here you'll find one particular insect. This is called as Aliroclava canangae, which is again an invasive one. But it is well present in our country, is what I understand. But now we are in the process of characterizing and publishing this particular Aliroclava canangae. This was first reported from Malaysia, followed from Indonesia. No report has come from India. We are in the process of now getting it identified and reported. So this is how the spiraling whitefly, especially the first one, which is the adults itself, you will find exactly. Nymphs you can identify by these two structures. This is very common in disperses. and you'll find the color of this whitefly is absolutely white. And this is how it is found in coconut. So how will you exactly identify it is based upon the structure of the vasiform orifice. So vasiform orifice, the lingula is smooth here. Sorry, the operculum is smooth here, but the lingula is tongue-shaped here. So this is a very important feature by which you will identify that it belongs to Alerodiscus dispersus. And you can find the compound pores. The compound pores are one of the very important features present in Alerodicinae. And you will find that in Alerodicinae, this compound pore will have a conical structure in case of Alerodiscus dispersus, whereas it will be totally different, a kind of a dagger-shaped structure in case of Alerodiscus ruchi operculator. So this is something about Alerodiscus dispersus. And this is no more a major pest in coconut, but this is reported as a very important pest in guava, and also in most of these ornamentals and vegetables way back in 200s. Now it has come down because of this natural buildup. And you will find ruchi operculators, and you will just find this egg. Here it will be complete spiraling of egg laying. Here it will be a kind of a split spiraling of egg laying. And you will find this nymph tufts as one hair. Normally it has this one fluff of hair rather than the other one which has two fluffs. And you will have the adult which has got the brown mottling. Here there will be absolutely no brown mottling. It's absolutely pure white. And you can see the colony, how serious it is. So this is quite specific to coconut. It's a very preferred post. And you will find the vasiform orifice. Just see here, the operculum is highly rugose. Rugose is nothing but the operculum is wavy in nature. Once the operculum is wavy, that is exactly the name it took in. It's called as Alerodiscus ruchi operculum. Operculum is rugose in nature. And you will find the lingula, which is acute and sharp. And finally, you will find the compound pores. Here, the compound pores has got a conical structure. Here, the compound pore will have a kind of a dagger-like process. This is how it is. And this is dispersus, and that is ruchi operculatus. And coming to the Bondar's nesting whitefly, and how we distinguish these Bondar in a single shot is the puparium will be flat here, whereas the puparium will be a kind of convex in the other one. And here you'll find both are. So the puparium features alone will not suffice for the identification of the nesting whitefly. So here the genitalia plays a very important role. So that is what I am trying to highlight. The genitalia is also equally important to get the clear identification. Now it is restricted only to the Para-Alerodus group, where the genitalia plays a very important role. Here the compound pores, if you just see, it is all a flower petal-like structure. So these are the compound pores available. You will find the compound pores here. There will be 4 bigger one, abdominal one, 2 smaller one in the abdomen, and there will be 1 cephalic also in the region. The same features are known for this also. So then how do you distinguish? So the distinction is the puparium is not in a position to distinguish between these 2 species, whereas the adult genitalia can distinguish. The adult genitalia has got 2 structures here in the top and the bottom. Here it has got a cock head-like structure. So in certain cases, the adult distinguishes very critically, and in certain cases, it is the other one around. So what we found in the coconut system so far. So though we had 5 whiteflies associated with coconut, they are all coexisting as well. We find only these 2 taking an upper hand. One is the rugo spiraling whitefly, which takes a bigger stride, and then comes your rugo spiraling, your Bondar's nesting whitefly. So they competitively regulate each other. Just see initially how they are flat up, and just see how they are flattened these days. And this is one of the reasons why these competitive regulation do happen when the insects coexist. And you'll find the parasitism by Entarsia is very, very critical. I read this is one of the very successful bioagent. And if this bioagent is conserved properly, because we don't have that kind of mechanism to mass multiply and release it in the field. So if you can conserve this bioagent, this can take care even up to 60% of the parasitism, and the population will come down. So that is one of the reasons why initially when the population shoots up in case of the rugo spiraling whitefly, the population, the amount of the natural meat is very low. Once the natural meat population shoots up, it gets stabilized altogether. And of late, this is reported very late. This is the 5th one which was reported on coconut. There is Alerotrachelus atratus, which is called as a palm whitefly. Here you can distinguish by the presence of the black puparium. And being a kind of a Alerocanthinae group, you will find the submarginal fold is very critical. Submarginal fold gets blocked here. It is not continuous as you see here. And you will find the initial fluff, the eggs are all black in color. And you'll find the lingula here, a kind of a knob-like structure. So this is a very important feature where you can distinguish all these things. So what we could make is, this is the one which is very, very preliminary to us. It was reported in 2003. You can just find Dr. Chandriya Mohan has also reported this is from coconut. And what you find here is this particular pest has an indigenous level parasite. We have very good predators like your lady beetle, anthochorid bug, cybocephalus. All these was in a position to reduce the population significantly. And this is one of the reasons why when the Rugose spiraling whitefly also came, these organisms, these predators, which are mostly general in nature, was in a position to suppress it to greater level. So I am very happy to say that the molecular characterization also has led to greater in-depth understanding of all the species. So morphologically, we could establish dispersers, Rugioperculators, Parallus bondari, as well as Miniae. Now, typically through the molecular characterization, we can form this Ailurotracheus attratus come under this bigger group. You will find Ailurotracheus attratus in this particular area. And you will find the dispersers and Rugioperculators coming in a separate group. And you will find Parallus bondari and Miniae coming in a separate. So, which means if you do the molecular characterization perfectly, you can really delineate the species very nicely. And that will result in the correct identification of species. And there are so many wrong deposits in the gen bank, so don't get confused. So whenever you deposit the right one in the right manner, that will lead to a good insight. So kindly do not deposit the wrong ones, or don't try to emulate what others have done it. Even there are errors at an international level. So kindly deposit the one which you get it perfectly. That will give a lot of insight. So we could exactly spot out that particular difference in terms of this Ailurotracheus flacosus. This is another invasive whitefly. This was reported in Goa. Dr. Sundaraj sir and team has reported that first. We could go into the adult features as well. You can just see the genitalia here. Just got a pointed structure here. The position of this point itself is very, very important. And what we found is we found some of these wrong deposits available in the system, and that led to initial confusion. We could get the correct identified nucleotide sequence deposit. Now everything is now becoming very perfect. So the humble request again is to go for the correct morphological identification, for which the experts are very much needed. Either Dr. Sundaraj or Dr. Dubey sir will be doing the greater level in this understanding. But at the same time, we should be in a position to correctly deposit this on. So this population has also come down very significantly these days. So this is this was reported in 29. This is Ailurotracheus flacosus, and this is the one which is reported known as the Pepper's whitefly, or the Chili's whitefly, or the Solanum whitefly. This is again reported by Dr. Sundaraj and team, and this has got a black puparium. You can just see it is here. The submarginal fold is very critical. Submarginal fold will not cut here, so that is a species where you can identify it. And here, as I told in Attratus, the lingula is round. The lingula is globular, or only a single. Here it is bifid. It is two different structures. So all these identification features, especially the correct, everything looks alike, unless and otherwise the correct features are spelled out. Now just see the genitalia of these two. The genitalia will be looking very similar, but here the position of the knob-like structure here is a bit far from the midline. So it is around 0.6, whereas the other one is around 0.3. So this is the one which makes the things very, very interesting in the genitalial identification as well. So this is the 8th one, which is called the tetra-ailurotus, which was reported from the leguminous species subopul. We could report this on Gliricidia, and you can find this U-shaped structure will be present on around this vasiform orifice. This is an important feature of identification. It's a black puparium, and you will find again the globules along the submarginal area. So these globules also make the identification very perfect. And one of my students who was at Madurai could identify one particular parasitoid. It's an Amita species reported for the first time in the country, and he has got the best poster award for that as well. So what I'm trying to highlight here is some of the features, especially the morphometry, are very, very critical. Why I have just put this is all looks alike, but the genitalia makes difference. You just see this thorn-like structure present here. It is 0.6%. So in Trachoides, it looks very much beyond the midline, whereas in Flacosus, it looks very closer. And you can also find the submarginal groove, which is continuous here. You'll find it continuous here. Here it will have a break here. So these are some of the areas where you can identify at the puparium level. Here you'll find the U-shaped structure and the wax globule, which is lacking here. And just see the genitalia of tetra-ailurotus acacia, it will be just rod-like, which is how most of these whiteflies are known. And we will find some of the setae, especially the 8th abdominal setae, are very, very important to identify it. So this is the one, sir, I would like to highlight here, because this is the latest one, which is not published. We are in the process of getting it published. And this is Ailuroclava canangae, which is reported as a pest on coleus, ornamental coleus. So we are at the process of it. And we could made this particular Aleroclava canningae, which is reported only from Malaysia or Indonesia way back, and also from US on the quarantine interception. We could characterize it, especially it has got a two-segmented setae, both two-jointed setae at the cephalic area as well as the first abdominal area. And your vasiform orifice is also very unique, and you have a very specific tracheal cup. And this is a very close relation with the Rusostigma eugene, which is present in Jamun. I can find how they are molecularly getting isolated between these species. So this makes it very holistic. And this is a general idea about all this, because time is running away. I'm just, I'll make it brief now. So you'll just see the diverse nature of this genitalia, which makes it perfect to get it identified. And we are in the process of even deciding how this genitalia will be used in the identification of the whitefly. And all these three and all these species are very much involved in this. And this is a kind of a female semen gland, we call. This is also one of the identification features as reported by one of my professors abroad, USDA, one professor Greg. He teaches all these two-shaped cup here. You can just see a snake head like cup here, smoother body. All these are features of semen gland associated with the female reproductive structure, which is also used in the identification to some extent. And coming to the biological control, I think coconut is always mother or father of biological control. Any biological control agent we have pioneers in getting in 1950s around Platymerus levicolis, assassin bug introduced from Zanzibar and for the biological control of it was not that successful. And the classical successful bug control agent is none other than the goniosus and your dracon used in the management of leaf-eating caterpillar, which is no more a major pest in the country. And we approach all these conservation strategies for the management programs because many of these natural meats are not in a position to multiply. So this is one of the classical examples to be very proud of. Our institute, our predecessors have done a greater level of work in this area. Just like COVID, the borders were all marked here. The agriculture department takes shape. They will never transport a kind of infested material across the districts. That was the precaution they were maintaining at that time. Even the DIPA Act was very successfully implemented in 1923. That was impact. And we had even both laboratory taking this parasitoid along all this area. Now, thanks to the State Department of Agriculture, they have seen that all the districts have got this parasitoid breeding station. And finally, the pest has virtually disappeared. So we finally, only in certain pockets, we have the pest now. So this leaf-eating caterpillar, which was a real menace, causing around 40 to 50% yield damage in most of the areas. And it takes at least 2 to 3 years for the complete recovery. Is no more a major pest in most of the coconut growing tracts. Thanks to the biology. This is how the success, it should come. And I think the very classic success story, which can be highlighted in the biological success story is this one alone. And you know, the Hans Saran, who has got even the World Food Prize, thanks to the efforts of promoting a biocontrol agent, that is Anagarus lopezi. I think now NPR has imported this particular Anagarus lopezi for the management of Phenacoccus manifestae. This is nothing but your cassava millibug, which is again an introduced pest of late. So this is the impact which it could create in Africa in terms of the pest management. And that is the impact which we could realize, but we are not in a position to project that in a very significant manner because this itself is one of the classical evidence of a successful biological control as he has done here. So in whiteflies, what we can do? So to our understanding, what we find is the whitefly dynamics depends upon mostly on the weather. So the 30 to 40% depends upon the weather condition. So once the temperature shoots up, it goes up. Once the rainfall goes up, it comes down. That's so when weather is a very important factor, no doubt about that. Now comes the Encarsia guadeloupe. This was introduced along with Alero dispersers. Thanks to that introduction, so that could make the way for the management of Rugi operculitis as well. And you will find the predators and pathogens 5 to 10. So rather than giving most importance to these predators and other things, our emphasis should be mostly on this. That is one of the reasons why we promote the conservation biological control is very important, because after 2 to 3 months, you will find these parasitoids taking upper hand in all these things. So this is the parasitoid. I will show some of the videos also. This is the predator. This is a cybocephalite, and these are the natural means. We have Simplicillium, Lanasonevium reported as entomopathogene fungus. Our colleagues from NPR has come out with the successful Isaria, Phimosaurosia. All these things are there, no doubt about that. But this biocontrol agent, especially the Encarsia and guadeloupe and disperser, plays a very important role in the biosuppression of coconut whiteflies in the coconuts, the this system, especially in the coconut system. Just see the emergence of this parasitoid from the parasitized piperium. And this is how the piperium will just find, we have found at least some, in a single frame, 12 to 15 parasitoids emerging from a single piperium. You can just see. So this is how the whitefly will emerge. And this is how the parasitoid, so parasitoid emergence would be around 4. So this is one of the areas, and you will find these are all parasitized piperium. And this is very clearly explicit in the field. And we have to see that they are not killed in the process of our intervention. Even neem oil more than 0.5% has a deleterious effect on these white parasites because they are very, very small in nature. So we have to conserve them. And we are using even the parasitized piperium strips to the newly introduced garden as well. So this is another important thing. I think I will give some credit, greater credit to our Chalapati Rao. He has standardized its mass production as well. So this also plays an important role, at least to the level of 1 to 5%. So this is the predation efficiency of a predator beetle, and this should be is not critically identified. So we have a kind of a confusion in the identification process. That's very important because this is not looking alike as Apatocrisa aster as described by Professor Banks. This is how Dr. Purni Madam highlights here. Here you can find this kind of markings is absolutely absent in the described one of Banks as Apatocrisa species, Apatocrisa aster. Here what we have got is a Apatocrisa species, and it needs a kind of a black mark in the antenna. This is absolutely lacking here. And the graded cross veins on the wings are green. Here it is black in color. So what I'm trying to highlight is the correct identification is very important. What we have identified and reported as Apatocrisa aster is absolutely not. It should be Apatocrisa species, unless and otherwise it is described greatly by us. And this is the cybocephalid, which is also playing a very important role, even in the management of your Bondas nesting whitefly. It feeds on the eggs. And so they make a very important, unknowingly, they are very tiny, small, and they play a very important role. And how are we going to manage this? Because seasonal wise, these kind of natural means take an upper hand. And also this has to be conserved. See, all these bioagents need to be either conserved rather than we augment. So which you have, should not avoid this pesticide spray is what our humble request is. So we have 2 bioagents very successfully evaluated. CPCR has come out with Simplicillium, Lanarsonium. This is also equally important. And we have from NBAR our ICAR-IIHR Fimosaurosia. So the whitefly population shoots up during the dry weather. So during the dry weather, this fungal pathogen has its own limitations, but still we can intervene in a way only when the population exceeds 20 expels. Then only we advise them. Otherwise, even the parasitoids take all these things. In the whole country, you will see these parasitoids that play a very important role. And if you can quote for that success, it is mostly because we have not applied any insecticide. At the same time, we have seen the parasitoids are conserved. And just see this particular thing. I'm very happy to say that we have identified a beetle, a leucornate beetle, which can feed on the sooty mold of this mold produced by this grown on this honeydew. That's very, very important. See, it is the black encrustation which makes the deterioration in the photosynthetic efficiency of the palm, but thanks to the beetle. So you'll find all the black material taken away during the monsoon phase. See, this is how the nature also makes an effort. So this was initially identified as Leucrodus. Now it's perfectly identified with Frank Schavaler, and you just see the leg of it. What I'm trying to say is this kind of mechanism is prevalent, and you should not intervene with any insecticide. So in those areas where insecticidal intervention has been made, especially in certain pockets of Andhra Pradesh, Tamil Nadu, still whitefly is a problem. So in Kerala, it is very much limited because of the nature of management they undergo. So we have evolved an indexing method and to see that how it can be scored, and we have a standard publication for that. What I'm trying to highlight, the dwarfs are relatively more susceptible rather than the talls. So the talls are relatively more tolerant. So in those areas, because our policy itself is now around 60% tall, 20% hybrid, and 20% dwarf. So promoting this tall is a great impact. That is why even when the whiteflies come, it did not make that kind of impact on the yield loss, though it has got some impact on the dwarf genotypes. So what I always advise to my team is to have a kind of habitat diversification. We should have all the crops together. This is the concept of One Health approach, which has to be promoted. So we have a small garden here. We promote all the intercrops, all the, we have a small kind of YouTube for that. Those who are interested, you can look. We have a bird perch there. We have a fish garden there. We have honeybee there. You name anything, we have it there. And we have produced different kind of volatile cues. And we also found a kind of more of this diversity in that. We found this garden reduced by whitefly infestation because of the diverse volatile cues which is being emanated from the system. And it is definitely climate resilient. It conserves the biodiversity and reduces the pest incidents, and it gives a continuous income to the farming community. So we are always promoting such kind of strategy, which not only reduces the pest incidents, but at the same time, it gives a continuous income. And it is climate resilient because the water will not fall directly on the soil. At the same time, the sunlight will also hit not directly on the soil. The soil temperature is above, it's always below the critical limit, and the microbes are being conserved. And we could recently identify some potential microbes associated with very good recycling of this material. And so we evolved a kind of the pesticide holiday conservation approach, need-based intervention by neem oil or even Simplicillium or even ICAR-IIHR are all advised. But our emphasis is mostly a kind of a conservation strategy to see that NCAR is better preserved in the system. So we found a meta trial, and we could find after 2 to 3 months, we'll find this conservation biological load equally effective compared with your ICAR-IIHR and the neem oil per se. So we have evolved a kind of a strategy. We have evolved a kind of a economic value for this conservation strategy, what we have made in the system. So we could say highlight around 17.59 billion rupees out of non-spraying insecticide, out of conserving the parasitoid, out of ecosystem service. It's a small projection which I have made, which is equivalent to around $220 million. So this approach, because of not resorting to a kind of insecticide application, and we have seen that this is being happened in most of the areas of the country, and the success is definitely very, very impressive to highlight and quote with. And the outcome is we got a value of so much of it. Again, my dear friends, for the last thing, wherever I go, I usually highlight this particular pest because it's a potential invasive which has not been reported in our country. So whenever you see, wherever you see it, kindly inform us. It will be attacking mostly on the crown portion, especially on the spindle leaf zone. This much of beetles are there. So it is a very severe problem in the Southeast Asia. It has not reached our country because we have 2 more leaf beetles here. Maybe these leaf beetles are not very problematic in our country, and the national means are taking care of this also. We do not know. So we will come to that. So the emphasis now is better soil, better plants, better human. So we have to focus more on the One Health approach, which not only reduces the pest impact, which also makes you more healthier and living major things. So finally, as a conclusion, we have to have a Paka diagnosis perfectly. The molecular characterization and correct identification is very important. Once you do that, you will find effective back-control agents for the effective suppression of the pest. And the readiness of biosecurity preparedness is very important. So as a part of a way forward, we give a lot of emphasis on this taxonomy, especially we are now giving emphasis on the endosymbionts as well. So biological suppression, especially to conserve, we'll see how best we can mass filter and release. And the loss estimation of these non-invasive, we are not doing very systematic in our country. We are focusing to do that as well. And the conservation of the biodiversity, because that's very important. Once you do that, most of your problems are solved. And this is what I thought I should share with you. And finally, before I conclude, in Tirukkural, there's a very famous message saying that better manure than plow, than irrigation, better guard. What he means is it is not manuring, it is not plowing which is important, it's manuring which is more important. And after weeding, it is not irrigation which is important, it is guarding. So that is how even in Tiruvallur he was emphasizing. So guarding is very important. If you guard it properly through the biodiversity conservation, I think we will go to greater level. And that is the talk of the day. And that is, I once again thank Dr. Sridhar for this wonderful opportunity. I think I've exceeded some time. I'm sorry for that. And thank you so much for this very nice opportunity provided. Thank you.
Thank you, Dr. Joseph Rajkumar. Before going for question answer session, we from the association side, we are extremely happy. We have shared whiteflies for farms, but you have given a glimpse of all the whitefly research and what happened in India. Not only India, starting from Linnaeus attempts of naming some whitefly, maybe in the wrongly with other insect, how it Karan Singh or Dr. David's contribution, that is up to Dr. Sundar Raj and others also who are doing. You gave a glimpse of different whiteflies, more than a dozen, which are hovering. Generally, when one invasive is there, what are the specific bioagents will be looking for that? When a complex of whiteflies are there, we don't know which whitefly is preferred by what natural enemy. In that situation, it is really the biocontrol interventions need special attention. Once again, in a short notice, we agreed. We are looking. This is because 125th month without a gap, thanks to the this online provision. That's how, without any gap, we could make 125 despite COVID interference was there. And I profusely thank, and we'll go ahead with the discussion with our permission. Very nice presentation. Compliments to your nice coverage. Yeah.
Thank you,
Now
Dr.
it is. Okay, thank you. Please unshare. Now we can have that open discussion. This.
Dr. Joseph Rajkumar.
Yes, sir.
Yeah, please.
Dr. Joseph Rajkumar, Ansarali from Kerala, Spices.
Yeah. Please carry
on.
Yeah,
very nice presentation, like UG class it is. We once again
now, yeah, that is true, sir. Now many postdoc scholars are here. I hope they got largely benefited from this one. Very, very informative. Please go ahead, sir.
But in MSc, we will avoid taxonomy class, but this taxonomy class is very useful for us.
Thank you, sir.
And also, what is the latest strategy? Any important, we can import any invasive parasite?
See, as far as our whitefly, especially the Rugio operculus is concerned, at the international level, there is one ICAR-IIHR noisy. Noisy has a kind of a blue scutellum, and that is not reported from our country so far. And they claim it is very effective. But ICAR-IIHR Guadeloupe itself is taking care to a greater level. So we should not give a kind of a competition among these native and the exotic one. So presently, no such attempts have been made, because even in Allirotractylus attratus, Dr. Selvaraj could identify one ICAR-IIHR cubensis. So that is also working, because that is not expanding the tentacles. So such very specific parasitoids are being identified, and they are taking care in a larger way. And it is our thing is to just see that they are not destroyed from the system, and for which indiscriminate application. I think I will be very happy, Dr. Answer, if you can share some of your Cincinella cardamomi also, because at international level, Cincinella itself is a big taxonomic concern. So all these things will add a lot of values into that. So we should look for these parasitoid, because for whitefly parasitoid is very important in the management. Thank you.
Dr. Joseph, luckily we have no whitefly incidents in cardamom for the past 3 years. Only scales are
very good. Very good. Because it's seasonal, because the weather factors are not very conducive for it. And that is well studied. I think Dr. Subramaniam and team, I think Dr. Sundaraj and team have studied well your Cincinella, because that's a native one. And that can have a lot of international significance. Why? Because the genus itself is named after Professor Karamsingh. So that is what the importance is. And second thing is, some importance is required in that. So if you can get it, I will be very happy. Okay, Dr. Answer.
Dr. Joseph, we recorded Ascarsonia on whitefly, Ascarsonia fungus. But what is the status?
Actually, Ascarsonia is not amenable for large-scale multiplication. So it has its own impact, because it's kind of an obligate, very specific. So in certain areas, Ascarsonia allirodis is used in the management as well. But we prefer that all these things has a better way of conservation. So that makes a lot of things. So it is not that one aspect. The whiteflies are managed by a lot of bouquet of natural enemies, and that has to be very critically analyzed, for which the indiscriminate use of insecticide should be avoided.
What about use of Pongamia soap or Pongamia oil?
Yeah, we found neem oil, and also see some of these entomopathogenic fungi by our NPR team as Iceria. We also have on Simplicidium. They are all working fine, but above all, the emphasis should be on these bioagents as well. So it is not that others are not working, but once the population exceeds certain level, here we say around 20 expirals. So that's very important. So that can be, if the population exceeds at that level, you have to intervene and then manage it. Okay, thank you.
Thank you.
Okay, Dr. Girish from Pune, DFR Pune. Dr. Girish, please unmute and
am I audible?
Yeah, you are audible.
Yeah, Dr. Joseph Rajkumar, sir, it was a very informative and nicely compiled presentation on whiteflies, especially even whenever we go for the survey and all, even I used to see a lot of different species of whiteflies staying together. So this talk has given me an opportunity to distinguish different in future if I go for survey and all. So I am working in the Directorate of Floriculture Research in Pune, and we also have a regional station at Kadiyam, sir.
Yeah.
We mainly on commercial floriculture crops, even the ornamental palms, the ornamental plants and avenue trees. These are all the major source of whitefly infestation. What is your suggestion for me to work on in this line?
Actually, Kadiyam, a word of caution to Kadiyam, I think the entire planting material production of the country is in that Kadiyam belt. Okay, I think you will not disagree with me. And the major thing is they produce a lot. Your planting material is firstly contaminated. The soil is contaminated with Melodogyne enterolopi. So that is another concern. That is a hidden pest and it goes unnoticed. Since whiteflies are quite explicitly visible, that is well seen also. So for these avenues, especially the ornamental things, what they do is, especially these ornamental, the nursery manager, they spray a lot of insecticides. So whatever way they do it, they spray it and see, they keep that they're very healthy and clean, because that is how they do the business as well. So we cannot come in their way. But coconut as a whole, no, it's a bigger thing where we don't advise. And here in the nurseries and all, we have to see that your planting material, anything for that matter, are absolutely free from any kind of pest before it is being transported. So for which all precautions have to be made. And for your information, how it has reached from, this Rugo spiraling whitefly from Kerala or Tamil Nadu to Kadiyam is simply because this Kadiyam nursery team was very much interested to have this COD coconut. COD is the Chavacard orange dwarf. Orange is very susceptible to this Rugo spiraling whitefly. So they make an exchange. How they exchange is, in Kadiyam area, they have got one Chanangi variety of their own material. So that they will be exchanging. Chanangi is a green dwarf, very specific to this Rajkumar tribe. And that material they have exchanged. So Chanangi will come to Kerala, COD will go to Kadiyam. So in the process, in this exchange process, they have not only exchanged the material, they have exchanged the whiteflies also. So that you should avoid, and for which strict quarantine, domestic quarantine should be implemented is what my thing. Otherwise, the planting material, everything you have, I cannot advise a kind of a conservation strategy for this kind of nursery. You have to see that it is absolutely free from these invasive whiteflies before it is being given to the farming community.
Okay, sir. Thank you, sir. Thanks for your suggestions.
Next, Dr. A.T. Rani from Chetali. Please go ahead.
Good morning, sir. Sir, I'm myself, Dr. Rani from Chetali. Sir, here we have this whitefly infestation, very severe infestation on all the horticulture crops here. So very severe in all the plants, whether it is avocado, rambutan, guava, whatever you take, whatever the plants we have here. So we have very severe infestation. Earlier, I also thought it is a Alurodicus dispersus. But now, after careful observation, and I posted the picture in entomologist group. So there I got identification that it's a Rugio percolatus. So how can we manage, sir? Because it's very severe here. And we didn't take up any we took only one chemical spray, sir. After that, we left it. So there is no use of spraying chemical. So we left it. It will go as it is. So, and we have observed black sooty mold growth even in the 6-month-old plants also, and 5-6 years old plants also. So how can we manage this one, sir?
Yeah, I think it's a good question. I was talking mostly from the coconut point of view, but these kind of material, especially your avocado, your guava, they are all highly seasonal, and you just see nature of these leaves also. They are all quite thicker leaves. So they are a bit susceptible to this Rugio percolatus also. And kindly look into the correct species level identification. That's very important. I suspect that it need not be Rugio percolatus alone. It can be even Bondari, Parallelis bondari, and as well as Parallelis minier. So that also you just try to identify. For the time being, what you have to do is you have to, if the population is exceeding 20, you have to intervene with some bioagents. You can resort to either very good neem oil spray to see that one round of that, and one round of your Isaria fimosorosia also, because these are all smaller plants which is amenable for spraying. You can resort to that, but see that your parasitoids in the older gardens, you will find the parasite that is also being conserved. So over a period of time, it will come down. Don't worry too much on that. But if you feel that it is exceeding, you can resort to bioagents, spray with a botanical or with an entomopathogenic fungus and reduce the population. That's very important because the population should not shoot to a greater level. And during the monsoon phase, we will be happy to give you our Leocrini beetle also. So that also, because Chetali is a kind of a forest area where you can have this kind of beetles release. So they will be in the system and your shooting mode will also be taken care of over a period of time. So you intervene, madam, no doubt in that. But see that you go for the correct identification. I still doubt it need not be your Rugio proculetus alone. It should have a combination with your nesting whiteflies also. Okay.
Sir, is it possible to give identification? Can we send a sample to you, sir?
You can send, but our main authorities are Dr. Anil K. Dubey, sir, because they are the real taxonomists who can vouch for the correct identification. To some level of my knowledge, I will definitely help you in that. You can even send some images. You are preparing images itself is enough.
Okay.
You can just send to me.
Okay, sir, I'll send the images of this whitefly, sir, then.
Yeah, you can send to me. Definitely, I'll be happy to do that. But the basic, because whenever you submit a paper for publication, they will immediately ask who has identified this insect. So the names of Dr. Sundaraj, Dr. Anil K. Dubey are very important there. Okay, so we have to get their services also in the correct identification. Okay, so
yes, sir. First, I'll send the images, sir. If it is not possible to identify, then I'll send the specimens, sir.
Yes, sir. That definitely we can do it. And you can, and also I suggest you, even if you can give a very harsh spray of water, just like what we go for a washing, kind of car washing, or even before painting, now we go for a washing. So such kind of washing, if you can, so if you have water, that's another question. If you can resort to, and you can see that they're displaced. Once their nesting colonies are disturbed, then actually the re-establishment, it takes time.
Okay. Thank you very much, sir.
Okay, thank you. Thank you. Next, Dr. Varghese, sir, you can.
Sir, you have to unmute, sir.
Please unmute. Please unmute.
Yeah. I thought I'll wait because I don't have many questions. I only I was the CPCRA-RAC member, so it was a kind of recapitulation for me, and exceedingly good presentation. And this is a kind of natural history presentation, which would stimulate greater entomological research and contribution to farmers. Just one or two experiences, Joseph. What we used to do when we in IIHR, I mean about 20 years ago, when we used to have the spiraling whitefly on guava. What we used to do, we used to go to the avenue trees in the city, Bangalore city, which was Michaelia champaka and all were infested, and bring those twigs and put it, and this Encarsia guadalupe would soon colonize, and that's a nice way of doing. And then we used to recommend it to farmers also. So that's a good thing, number one. Number two, the conservational biological control with diversity in crop ecosystems, I think in the RSC also addressed it. Suppose you take a place like Arsikare, Tumkur and Tiptur and all, which is a typical Karnataka belt of coconuts, how to get them to do it into that kind of a mode is difficult because they are a monocropped site, So for that, we need to kind of get into some kind of other management strategies. But biological controls, but conservation is excellent in Kerala. My own cousins and all that, they all practice that in Kerala, and they have found it very good. But these areas also we need to put some thought. How else? How can we have some creepers growing or all that? That kind of thing. The third and the last point is, how do we forget the domestic quarantine? We, apart from that, the first port of entry, say maybe Trivandrum, maybe Cochin, maybe Calicut, have they come through that? Or have they, we always assume from Kerala it has gone northwards. So it can be the other way also, or it can be east-west, can be from Chennai, can be from Bangalore. But at the port of entry, we all have quarantine labs. So how do we enhance their handling capability? That is one thing which is of concern. So maybe we should also get involved with the post-entry quarantine scenario. These are my three kind of remarks. Thank you so much. Keep doing well.
Thank you, sir. Thank you. Thanks a lot, sir. Sir, the last point which you have mentioned, I always feel, sir, we have been updating our quarantine team also, because even whitefly, simple piparium, if you can put in your pocket or somewhere and take it, no, that is enough, because they don't. So that kind of awareness, even our own people should feel it, because normally in India, the domestic quarantine, yeah, they take material here and there, okay, but even those people who are visiting abroad, no, whenever they see a very good ornamental plant, no, they try to bring it to our country. Whether they follow this protocol is a real question mark. So that, because this is definitely not a billion market. So even if passenger carries a plant material, that will not come to the limelight. Whereas if passenger carries what is it, golden biscuit, that will be in a limelight. So everybody will be focused on that. So that is exactly how it is being traded. And there are, sir, some illegal modes of now transfer as well. That is where we have to intervene very strictly, because a lot of invasive, I mean, exotic fruits are now coming into the country, and they are one of the best carriers. Now you find a lot of different varieties of your rambutan, your jack, your avocado, everything are coming into the country, not through the right mode, is what I feel. So if that areas are, because they don't pass through the strict quarantine group. So that is very important, sir. So that is absolutely in the Ministry of Agriculture, and we have only a technical say on that, not on the command of a law imposition on that.
Yes. You're quite right. Yeah.
Yeah, thank you, sir. Thanks for this useful discussion. Yes. Dr. TV Singh, sir, just please.
Yeah, you see, I also joined Dr. Varghese. I don't have any questions. It was a very informative, lecture, informative talk. Congratulations to you, and congratulations to the speaker. And congratulations to you and the entire team at Indian Institute of Horticultural Research for going to bring out a beautiful book on all the abstracts. Keep it up.
Thank you, sir. This is already, it is put in this Zoom link only. Those who
I am on a mobile just to.
Okay. Those who didn't see that one, are those who joined late, just put mail to aapmhg@gmail.com. Immediately we'll send the copy. It is thank you for joining from Hyderabad, sir. Here one of my batchmate, he joined from Orissa, Bhubaneswar. Dr. Mishraji, how are you? If you are online. In the meantime, any other
good morning
would like to? Yeah, how are you, sir?
Good morning, Sridhar. I'm fine. I was just going to the talk of Joseph Rajkumar.
Yes, sir.
Yeah. It's very informative. Very informative for me, because we are all retired. Still, I have interest, so I join many times this talk. And Dr. Sridhar has already told that he has shared all the previous talks, almost 155. So that I will also go through. Please carry on. Good work.
Thank you, sir. Thank
you, sir. Anybody else? Yeah. Otherwise, now it is time to thank once again that Joseph giving a complete idea. Those who are already having an idea, it is a feast for them, particularly those who are working on biocontrol of these invasive whiteflies using biocontrol. For others also, it is an eye-opening type. Because even I was last month, I got one PhD thesis wherein I got a coexistence of several whiteflies. I was not convinced. Then I started going through the literature, and that is possible. Then I have gone forward, because it was mentioned not just two species, more than two species coexisting. That is possible, what we have referred also, and it is a very comprehensive work. What you have not done is, only thing you have not mentioned the insecticides, because your emphasis is not to go with the insecticides. I have only one just a clarification I need. Whenever the population is too high, one of the recommendation is first to go with one or two insecticide sprays, followed by biocontrol, so that it will be more effective. Whether that is good or not at all, go opting for insecticides. That is better in this scenario where we have highest incidence of some of the whiteflies like Rugos?
It's a very interesting question, Dr. Sridhar. What I only feel is in coconut, we have at least 36 productive leaves. Okay, the fronds we call 36. So even if 2 or 3 are badly affected by this whitefly, I think it can be rejuvenated in a period of 3 to 4 months. So that creates a niche for effective multiplication of the parasitoids. That is number one. So if you spray an insecticide, what happens is the entire scenario is lost. Entire parasitoids which are developing is lost. It takes some time in all the areas. So in coconut scenario, I always feel that it is very harmful to go for any insecticidal spraying. One thing, in most of the areas, it is a kind of a homestead crop where the human beings are badly affected. Second thing is, coconut and the pollinators are inseparable because that's a very big area. So pollination also will be affected. So I feel that even if a farmer loses 1 or 2 leaves, he can recoup it back, but rather than avoiding a spray. But we can definitely go resort to some kind of these botanicals or even some of these entomopathogenic fungi. They do work well under such kind of condition, but kindly avoid the insects because that is not going to make that kind of impact in this whitefly scenario. I'm talking only with regard to the coconut.
Okay. Thank you. I got some practical points to be noted. Thank you.
Anybody else? One quick
please.
question. Those any of these spiraling whiteflies are vectors?
So that's another important area because you see, whitefly, even a single whitefly, we never allowed to sit on cotton because it's a vector of leaf curl virus. So that is one of the area I think everybody is now worried about. Now there are reports that this Begmo virus is being transmitted even by Ailerotrachylus stracoides, which is called as the Pepper's whitefly. And there are very few reports on this Rugio operculus also coming because we have not recorded anything on this coconut. We are at it, sir. But so far, because it has found to be quite severe also in some areas, we never found any kind of symptom expression on the coconut per se. And in all the areas where it was previously infested, now it is completely cleansed and they're looking very hale and healthy, right from south down to the northeast. There are reports on the vectors as well.
Okay.
Right. Okay. Now, I think everybody is satisfied with today's scintillating lecture. He's a very dynamic entomologist. I know him closely. We actually just before COVID started, that week when the COVID incident started, both of us were in Tamil Nadu. That seminar was on invasives. I was speaking on Tuta. He was speaking on these invasives. When I called him last week, that was the incidents I recollected and I asked him, is it possible to give it on 29th? Because before the end of this month and that to end of this year, on 25th month, he readily accepted and gave this one. Once again, thank you, Joseph. I thank all the participants. In the last 150 highest, I think, who gave companies that Varghese. That's how in the abstract book, we thanked him for attending and supporting these talks. And Dr. Mishraji also. And then I said, Dr. TVK Singh, every time if he is not traveling, I will be joining or I will not be able to join. He used to send that message. And naturally, when you are organizing something, this for the purpose of sharing knowledge in plant production, when we are doing it for, when we did it for 125 months, it is a satisfying moment moment for our group. And I thank all the participants. Here physically also several students are there because IHR started giving under IARA have MSc and PhD, several have joined. 63, we started taking classes since last 2 weeks, pathology as well as entomology. Majority of them joined here. I hope now onwards, as they will be having an opportunity to have this type of lectures, I hope it will be useful for them too. With this, I thank each one of you and Dr. Varghese, Dr. TVK Singh, Dr. Harprasad, Mishraji, Dr. Girish who joined, and Dr. Ali, and each one of you. I am unable to see all the names together. I thank each one of you, and a special thanks to today's speaker, Dr. Joseph. And we'll be joining with a next next talk in next month and for 126th month in the new year 2024. Happy New Year to one and all. Okay. Thank you so much.
Happy New Year.
Happy New Year to all.
Yeah, thank you. Same to you.
Thank you. Thank you.
Thank you.
And, that Joseph. Yeah.
Yes, sir.
We are joining from your lab.
Yeah, I'm joining from a lab. Yeah. I came down yesterday. So
right.
that's why I told, no, 29th will be convenient for me.
Yeah, you are out of station.
Yeah.
Right.
Thank you. Mr. G, how are you?
Thank you so much.
I'm fine.
Okay, because I'm seeing you after a very long period of time.
Right.
Okay.
Actually,
So,
many of the even Sridhar's talk I could not attend due to